fitness should predict longevity, right? It just makes common sense. If you're more fit, you should live longer than people who are less fit. That may be true up to a certain point, maybe 90, 95 years, but where are the fitness enthusiasts at 115? Where are they?
Welcome, everybody. This is Buck Joffrey with Longevity Roadmap. Today, I've got an interesting one for you. Dr. Michael Lustgarten. Now, this is an interesting guy. He's a PhD, follows the role of the biome in the aging process and, the gut muscle axis. This is something new to me. We know the gut plays a big role in health and longevity and health span and all that, but he's got an interesting take on it. He's also really interesting because he is an actual biohacker. Now, this is for me, the word biohacker is not necessarily a very positive one, because I think of all the influencers out there who, not all of them, but a good chunk of them, a good percentage of them really are just voodoo people, right? Like they're just telling you to do this and take 100 pills and telling you that their biological age is like 30 years below it is and that they're going to live to 150. Anyway, you know who the people I'm talking about is. This guy is a biohacker, but he's a scientist. You'll notice in this interview that he goes into enormous amounts of detail. But the reality is, if you're going to be a biohacker, you want to be more like this guy. And the idea here is that he's doing interventions, but he's tracking them. He's trying to see if they make a big change, right? So anyway, this is a fascinating interview for multiple reasons, obviously from the gut standpoint, but also if you're interested in this whole gut biohacking space, I'd encourage you to follow his lead rather than some of the others out there. And we'll have that interview right after these messages.
Hey, longevity enthusiasts, it's time to take it to the next level. I've been fine-tuning my longevity regimen for years, and I look better and feel better than I did a decade ago. In fact, my blood work is even better than it was back then. And it's all because of my data-driven regimen, and it's inspired me to create a course and community just for you. It's called the Longevity Roadmap, and I urge you to check it out. If you're tired of your belly fat, tired of being tired, or just want to optimize yourself for the next 50 years, visit longevityroadmap.com. That's longevityroadmap.com.
Welcome back to Longevity Roadmap. Today's guest is Dr. Michael Lustgarten. He's a scientist who is, he's in transition, previously a professor, and now going to Austin to continue his work that focuses on the intricate relationship between the gut microbiome, serum metabolome, and muscle health in older adults. He's got over 25 peer-reviewed publications, more than 2,400 academic citations, brings this rigorous scientific lens to aging and applies the same rigor to his personal health. And he's widely known for his meticulous self-tracking approach. He's documented this through his YouTube channel, which is Conquer Aging or Die Trying, where he shares detailed analysis of biomarkers, diet, sleep, exercise, and aging. He's also the author of the book Microbial Burden, a major cause of aging and age-related disease, where he lays out a compelling case for how chronic microbial exposure may drive many of the underlying mechanisms of aging. Welcome to the show. How are you, Michael?
Hey, Buck, quick correction. So papers are up to, I think, 33, 35.
Oh, man.
Citations like 4,500, but that stuff.
We're just outdated. The internet needs to get updated, my friend. It needs to get updated.
Whenever someone's like, oh, my H index or how many citations, I'm like, people are still dying. Doesn't matter.
Yeah.
All that street cred doesn't matter until death is eliminated. Now that's the big street cred.
There you go. So, you've made a pretty big name of yourself, not only in academia, but also sort of in the biohacking world. So what got you interested in this space? And then on top of that, I guess, why did you choose to focus, your perspective, the gut, serum biomarkers, and muscle health in particular?
Yeah. So it's kind of a long story. I'll try to keep it, as condensed as possible. But I've always been interested in optimal health and fitness, lean and ripped. I grew up on bodybuilding magazines when magazines were a thing as a kid. Yeah. So, Joe Weider and all those guys, in the bodybuilding world. So I grew up on that. So how can I be in elite physical condition? That was always my mindset. Fitness has always been a part of the approach, but it wasn't until my 20s when I had to figure out what I wanted to do with my life, and I really had no idea. So at 27 or around 27, I read a book by a famous calorie restriction researcher, Roy Walford, who the book was called Beyond the 120-Year Diet, which essentially detailed, I don't know if you've ever heard of Biosphere 2.
Yeah, I think I know what it is, but you can
So
explain.
it was basically a group of scientists living in a self-enclosed dome
Right.
somewhere out in the desert, trying to simulate conditions on Mars to see if they can actually live in a dome, grow all their own food and all that. But anyway, they had some unforeseen circumstances. They weren't able to grow as much food as they wanted. Everybody ended up calorie restricted. So Dr. Roy Walford wrote a book detailing the biochemical changes that went on, during that time, how a whole bunch of biomarkers improved and which ones that they were. And then he detailed the whole science of calorie restriction up until that point, which there was an abundance of literature, even, in the 90s and early 2000s, in terms of it being the gold standard for extending lifespan. So as soon as I read that, I knew that's what the light bulb went off. So went back to school, focused on biochemistry. My first undergrad was English literature, so I had no idea back then. So the second time around, it was just diving deep into the hardcore biochemistry. I wanted to understand what is the signal and the signals in our blood that are aging, right?
Yeah.
And based on the premise, aging and disease, they don't happen overnight. These are biochemical processes that happen over a very long period of time. So if you know what that looks like in youth, you can track it, you can potentially optimize it, you can slow it, you can flatten mortality rates, or you can potentially slow mortality rates. So, as slow as possible. So yeah, I went to grad school, focused on physiology, and then started my career as a scientist, still doing this stuff somewhat on the side, just the yearly physical, logging it into an Excel file. But it wasn't until 2015 when I really kicked it up a notch and I was like, I need data. I don't know. There's a scene in The Matrix. I don't know if you've ever seen that, where
Oh yeah.
Neo says, guns, we need a lot of guns.
Yeah. Yeah.
And I had that moment only for data, 2015, where it went off. And I started testing more often, logging my diet, recording it in a spreadsheet, and then lining up the diet, maybe even supplements with the blood test data, looking at correlations, and trying to push all of my biomarkers to the most youthful state, like not just metabolic health, but kidney, liver, immune, the full spectrum of biomarkers. So that's where we are now.
I guess what led you to actually focus on this through the lens of the gut?
Yeah. So after getting my PhD, and I know it sounds so pompous, I'm not trying to sound pompous at all, but after I got my PhD, I did a postdoc here in Boston, and that was focusing on the serum metabolome. So essentially, as many circulating metabolites as you can detect in blood in association with muscle mass and function in older adults, based on what are the mechanisms, what can we learn more about the mechanisms underlying sarcopenia during aging. So we published a bunch of studies, associating metabolites in blood with muscle mass and function, and postulated mechanisms or novel mechanisms for what may be going on. And a dominant signal in that pattern were gut bacteria derived metabolites. I didn't care about the gut at all.
Yeah.
So, and it was paper after paper, we saw that signal. So I wrote some grants, got the grants funded, did a couple studies on looking at the, gut microbiome differences in people who are high functioning, low functioning. And back then, 10 or so years ago, the idea that the gut could impact muscle wasn't a thing at all. And there was very little data. Now there's an abundance of data that it can.
When you were talking about those signals that you were seeing, I guess just from the metabolomes and following those, what were those correlated with? Like less disease, less chronic disease? What did you find that correlation with initially?
Yeah, so we were focused on muscle mass and physical function.
Oh, okay. Got it.
Whether it was lean mass, whole body lean mass, or appendicular lean mass, arms and legs, or thigh muscle compositions. You had CT scans in people and looked at, how much normal density and low density muscle, and then also measures of physical function.
So that's where I guess this concept, which, I hadn't really heard of before, gut muscle axis, that is presumably something you've coined. And talk a little bit about that. Talk a little bit about this gut muscle axis.
So it existed before me, not very much though, but we have contributed. There are a lot of labs who are doing great work now, really digging into it. So I wish I could say I was more convinced about the existence of a gut muscle axis in young or in old, but because we recently published an animal study where, all right, so the long story short is, can you optimize conditions in the gut to produce certain metabolites, for example, short chain fatty acids, which can impact muscle mass? So if you grow mice without a microbiome in a bubble, they have less muscle mass. And when you add bacteria-derived metabolites, short chain fatty acids, which are produced by soluble fiber fermentation, the germ-free mice that don't have a microbiome, it increases their muscle mass and their function. So that suggests that if you have a high soluble fiber diet, you have more of these short chain fatty acids being produced in the gut, and they can positively impact muscle mass and function, at least in mice. So we tested that in aged mice. We fed them a diet that was high in soluble fiber, 3 times higher than the standard chow, which is like the gold standard of diet in animal studies, for better or worse. So 3x higher soluble fiber. And as a group, the male and female mice didn't have higher levels of muscle mass or muscle strength, but treadmill endurance capacity was increased. So there may be a role for improving muscle endurance. And then we recently did a similar human study where we just completed enrollment and we're analyzing the data, where same story, we've got 2 different groups, high soluble fiber diet and, lower, not a low, but the USDA guidelines around 30 grams total fiber, 5 to 6 grams soluble fiber. And I don't want to say which way the data went, but preliminary analysis before it goes to the biostats core is it doesn't look like there was a major difference on muscle mass or function. So I'm a bit skeptical. The animal data is what it is, and that there's pretty strong animal data that there's a gut muscle axis, and there may even be localized situations where the gut can impact muscle. For example, if you've got a really bad, quote unquote, dysbiotic gut, maybe that can impact muscle mass, or antibiotic treatment. Maybe that's bad for muscle mass. That's been shown in animal models. But in terms of generally healthy people and jacking up fiber, soluble fiber specifically, we didn't make a dent.
Yeah, interesting. Well, it's good to get that data because, I mean, obviously, you look at, the number of animal trials that produce anything really positive across the board. Look at the ITP in terms of like the mice and, these, just for people out there, essentially looking at individual drugs and how they affect the longevity of mice. I mean, there's some that have come out and clearly dominated there, like rapamycin and that kind of thing. But there are others out there that we seem to think might have a benefit in humans, like metformin, and there's some clinical evidence that might have some protective effect, but it didn't show that in the ITP. So anyway, mice and humans are not the same, although it's, it's a useful reference point.
But wait, along those lines, as you know, there's a whole groups of people who are taking supplements and prescribed medications based on like ITP and other similar studies. So I'm not opposed to that, but what I do encourage is testing. Like, don't just assume that even interventions in people, RCTs in people where something may impact a certain phenotype, certain biomarkers. There's a huge group of people who will say, oh, it worked in them, I'm going to do that and it's going to work for me. And then don't test themselves. They have no objective data. It's just basically hope. So one thing I try to promote heavily is test. We don't have to wait for studies in other people. Test yourself. See if rapamycin or this intervention or that intervention, what does your data show? And if the data isn't good, take it out and put in stuff that actually is good.
That's an interesting idea. In a lot of these supplements, though, I feel like, I don't know, I take rapamycin, and I don't know that I could elucidate whether or not it's making an impact when you consider all the other things that I do, So anyway, but yeah, it's a good point. And that's one of those things too, where I think part of the problem in this space is that, like, there are some influencers out there that are basically, telling you to take everything and, bragging about how many pills they take a day. And that doesn't help either. I mean, that could be detrimental as well. You had a study involving transplanting human gut microbiota into mice. And what did you find about causality of, a gut muscle relationship there?
Yeah. So we took stool samples from high functioning and lower functioning older adults, and then transplanted that into mice that didn't have a microbiome. And there's a bunch of studies where if you do that, you can replicate most of the microbiome from the people into the mice, because the mice didn't have a microbiome. They're basically, at least in the short term, it replicates. So when we did that, there was a No impact on muscle mass, no impact on endurance capacity, how far the mice could run, but they did have a significant improvement in grip strength. So the mice that got poop from the high functioning people had better grip strength than the mice that got poop from the lower functioning people. Now, we measured a month after we did the transplants. An argument could be made if we measured a week, we may have seen changes in muscle mass and function. I'm guessing that the microbiome may have adjusted over time. And then there were other factors that may have affected the overall results. So the biggest bang for the buck might have been looking at 1 week, but academic studies are so limited by budget. You can only look at 2 time points and don't really get the whole story.
What's your thought in general? I mean, I know there's a lot of people out there who are really interested in just like human fecal transplants for, I'm guessing you know a little bit about that. Tell us a little bit what your feelings are about that.
Yeah. So the FMTs is kind of the same boat in terms of supplements for me, where I'm more interested in discovering one's personal recipe for optimal health, diet, exercise, sleep, not just, I'm going to just eat a certain amount of calories, but how would you even know how much protein, how much carbs, how much fat, how much fiber? Should it be 60 grams of fiber, 120 grams of fiber, 30 grams of fiber? Where does your data look the best? So when it comes to FMT, I'd say first, what are the conditions in your gut microbiome? What does that even look like? And just knowing who's there doesn't even tell the story. It's kind of like the human genome story. You can get your full genome sequence and say, well, your genetic risk for this disease or that disease is X. But that's what can happen, not necessarily what's happening. So in order to know what's happening, you've got to look at end products of metabolism or metabolomics, which is why I focused on metabolomics. So for the FMT, I'd say, what is someone's microbiome? And even then, if it may have an increase in quote unquote bad bacteria, things like enterobacteria, and low levels of the short chain fatty acid producing bacteria, which are quote unquote good bacteria, you'd want to actually measure things in blood, whether it's the short chain fatty acids or other metabolites. And I can list a bunch, but you can measure their end products in blood. And then if that's the case, I wouldn't go the FMT route. I'd see, well, okay, what is it a part of my diet or supplements, or is it just not enough exercise? Like, what can I do to address the root and the foundation of my house before jumping to some exogenous agent without addressing the root?
So let's go back to this metabolites, right? I mean, you've analyzed hundreds of, now thousands of them in relation to their benefit. What are maybe are the strongest predictors of longevity or mortality or even just chronic illness?
Yeah. So actually, some of them aren't gut bacteria derived. I have a video on my channel yesterday that uric acid, and this is only in a panel of 12 biomarkers, but total cholesterol was there, glucose was there. So relatively low uric acid was associated with an increased odds of reaching 100 years. Whereas the other side of that, having uric acid levels greater than 6.5 milligrams per deciliter, the odds were significantly lower. So that's probably a kidney function story, because creatinine, which increases during aging, it's a kidney function biomarker that was associated with higher levels were associated with the reduced odds of reaching 100. So most people aren't thinking about optimizing kidney function, and that's a big part of the longevity story.
Yeah. Uric acid is one that is totally underappreciated and has a pretty significant role. A lot of cardiologists are looking at it now as a predictor of cardiovascular disease. By the way, one of the drugs that I actually take, it's not very common that people do this, but is a drug related to gout, colchicine. There's some really good data for colchicine in people who've had myocardial infarction, have heart attacks, and not having repeat ones. It seems like the best way that we know of, at least right now, to reduce cardiovascular inflammations, namely HSCRP, high sensitivity CRP. Let me ask you though, like when you talk about the metabolites, compare them with traditional biomarkers that we're talking about, like, HSCRP or stuff like that we normally are looking at when we're trying to predict biological aging.
So I see a hierarchy to optimal health and potentially longevity. At the top of the hierarchy is body composition, and not just how much muscle mass you've got, but body fat. And then body fat's important because it's proportional to visceral fat. Visceral fat, as you know, the fat that surrounds our organs, increases during aging. And why that's important is because if you remove visceral fat from rats, they live longer. So keeping visceral fat low, youthful, relatively youthful, not high levels, but relatively low levels, and optimizing body composition, resisting the age-related changes for lean mass, muscle mass, and bone. Most people aren't thinking about bone too until it's too late. You fall, you break a hip, the bones were porous, and they weren't thinking about it. So body composition is top tier. Physical function also top tier. So
yeah. So you're basically talking about lean muscle mass and VO2 max, which, those variables are probably the two biggest predictors right there of longevity, right?
But there's some nuance to that, right? Because I see a lot of people in the health and longevity space chasing the higher is better for VO2 max story. So when you think about what's the ROI on life expectancy increase or association with VO2 maxes, and we're not talking about 60, this is anything greater than 40. It's only 5 to 7 years, which is perplexing because how can you, fitness should predict longevity, right? It just makes common sense. If you're more fit, you should live longer than people who are less fit. That may be true up to a certain point, maybe 90, 95 years, but where are the fitness enthusiasts at 115? Where are they? And this is the same story also in animals. And I'm pro exercise. I've got my, and I stay lean. So in animal models, exercise there too can increase average lifespan, but it doesn't increase maximal. So there's something about exercise that is obviously great for health span and average lifespan, but doesn't get one to the maximal. So
Sure.
I'm not in the as high as possible for VO2 max. What could be a part of that story that may limit getting to the maximal lifespan? So I think most people that train, so of the 50% of the population that may train, most of them are probably overtrained. Whereas the 50% of the population that don't do any exercise, probably undertrained. But for the people who are into fitness, how can you titrate the exercise dose that maximizes your cardiovascular and other aspects of fitness, but you're not overtrained? Because if you're overtrained, you're going to have higher levels of inflammation. That's going to be bad for metabolic health and a whole host of other things. So I use HRV and resting heart rate. I use that to guide, my duration of workouts, intensity of the workouts, frequency of the workouts. Now, does that mean my VO2 max is 60? It's not, but I'd rather have my metrics of overtraining not look chronically overtrained, which is what they did before I started on this journey, and have as high of a VO2 max as possible without the overtrained phenotype.
Yeah, right. Absolutely. And, some folks have described that concept as sort of the Goldilocks zone of exercise. You absolutely can over exercise. And if you do, it's related with more cardiac fibrosis and atrial fibrillation, any number of things. So it's useful to make sure that you are not thinking that you are just going to exercise yourself into longevity by just going crazy. So,
but average longevity, and it'll improve health span related measures, but like you said, all of the other risk and conditions that may come with that. All right, so wait, that's top tier.
Yeah.
Without a doubt, that's the non-negotiable. Like it's terrible because when I see people in the, even scientists talking about longevity, I'm like, you can't outrun that foundation. That's got to be the foundation for anybody serious about this, right? So, all right, but then, I'm sure you know the name Jack LaLanne.
Yep.
Yeah, so that's the Jack LaLanne approach: eat real food and exercise. But that's actually a big part of what got me into all of this way back when. There's got to be a higher level. What's the higher level than eat real food and exercise? So what's under the hood? What are the biological mechanisms that underlie body composition changes and functional changes during aging? Well, so now we can look at a blood sample, right? And, we can identify a lot of things going on in blood. But just to take it from the top, you've got cells, proteins, metabolites. So looking at red blood cells and their related measures, the size of the red blood cell, which provides value because they increase during aging. So trying to make sure your red blood cells are not, getting bigger with time. But even the distribution, do you have a lot of small red blood cells, small, medium, and large, or are they all large? That's the RDW. That's a big predictor of biological age and inflammation and a whole bunch of other adverse health-related conditions. So hemoglobin, which declines during aging, oxygen carrying capacity. So red blood cells and optimizing all of their related parameters, big part of it. And note that red blood cells are the most abundant human cell type in the body. Most people aren't thinking about it at all. So then you've got white blood cells, and not only looking at total white blood cells, but neutrophils, monocytes, and lymphocytes, which are about 95% of the total. And it's important to look at those individually because white blood cells they're not homogeneous in terms of age-related change. The neutrophils and monocytes increase and lymphocytes decrease. So if you're only looking at the total, it may be flat during aging, but the distribution changes. So the neutrophils, monocytes, lymphocytes, also platelets, which are a part of immune defense. So now we've got the cells in blood. Now that's 45% of the blood volume remaining in that plasma or serum. Now you've got the proteins, metabolites, lipids, etc. So most people in terms of those metabolites, they're thinking metabolic health, glucose, lipoproteins, because they're at a relatively high concentration. So if we look at the biggest trees in the blood forest circulating, it's albumin and globulins and fibrinogen. In terms of concentration, they are far higher than glucose. Glucose is 80 milligrams per deciliter. Albumin is in youth 5,000 grams per deciliter. So, if you're looking at glucose, it's a small but important tree in the forest, but albumin is a giant tree in the forest. So looking at the biggest trees in terms of proteins, albumin, globulins, and fibrinogen, but even the globulins can be subdivided, IgG and then specific IgGs. So then now we're in the metabolites, right? And then looking at, sugars like glucose, the lipoproteins, but then taking that a step further too, which is the metabolomics, what I specialize in, which is looking at things that are on the cutting edge of, for example, kynurenine divided by tryptophan, which is a marker of inflammation. It's associated with neurodegenerative measures. Nobody's thinking about that, but that's going to be a big 2 biomarker list going forward. And then some of these gut bacterial metabolites that are associated with poor gut health that increase during aging, indoxyl paracresol sulfate. Now you could even go below that in the epigenetics, but the epigenetics too is kind of like the, and I measured like 17 times. So I'm not, new to that space. I'm very familiar with what it's measuring. The epigenetics are based on real biomarkers, and it's a prediction of the real biomarkers. So I want the real biomarkers. I don't want the epigenetic prediction of those biomarkers. So it's looking at all layers, it's proteins, lipids, sugars, all the metabolites, or as many as possible, the cells, white blood cells, red blood cells, and then going up to the highest tier, body composition, physical function, and regular tracking for all of it, not one test a year, but as many tests as one can manage, in their veins and fingertips to collect data.
So obviously, you've been talking about a lot of data, and I understand, you're using AI, machine learning, and various statistics to optimize your own data. But how do ordinary people do some of the things that you're doing? Like people are listening to this and they're like, that's great. And what am I going to do with this? Because I'm not going to do what he does. I'm just curious, like, if somebody's interested in doing more, but I don't know that they would even know where to start with the kind of stuff you're talking about, what do you tell them?
Yeah, so I offer consults, so we could start there. I offer consults on Patreon. I'm not cheap though, but this is high level stuff. So that's one. Two is I'm still using very simple statistics for better or worse. Just unadjusted correlations can go very far. Like if you've got a bunch of blood test data and track dietary data, you can just plug that into an online calculator and generate a correlation and then see if given food or diet or whatever is associated with a biomarker. So I started off just using Excel, just generating those correlations in Excel. Now, there are layers to that I've added, adjusting the models for body weight so sure. Now, with that in mind, we're working on an app, and that's coming around the corner very soon. So if someone's got blood test data and they track their diet, they can enter all of it, and then we'll give them the correlations, what their data shows, and then, go after the top correlations to try to move their biomarkers towards youth. So the easy answer is I offer consults. I work with clients. The other is, we've got an app coming where anybody can just enter their data and use that on their own.
One last, I guess, question in terms of your philosophy. And it seems to me that, you're tracking a lot of stuff, and even on the immune side, just having the fortune of interviewing a lot of different folks. I interviewed somebody who's sort of leading the charge from the Buck Institute on immune aging. A lot of this stuff has got something behind it at a higher level that's trickling down in what you're seeing as the products, the metabolites and that. Is your position or is your approach to try to influence those downstream metabolites one by one? I mean, I'm just curious, kind of like, how do you do that? Are you you're talking about looking at, all the little different elements of the immune system and trying to influence one by one. Is that kind of what you're doing?
Yeah, so it's easy to get lost in the details, but I try to focus on the biggest fish. So, you can subdivide lymphocytes into B cells, T cells, and then you can even further subdivide those into based on the surface proteins that they're expressing. I don't go to that depth yet. I try to keep it broad, general, neutrophils, monocytes, lymphocytes. So if you look at one biomarker in isolation and say diet associated with glucose, sure, you can optimize your diet to optimize glucose, but then does it impact kidney, liver, immune, etc.? So after every blood test, I've got basically a spreadsheet of, the correlations, all of the diet, macros, micros, foods, and then I've got 25 to 30 biomarkers going across. And I then take the score for each of those inputs. So how is omega-3 correlated with each of the 25 to 30 biomarkers? And then I go down the line for all of the inputs. So for omega-3, it'll have a score. And, is that biomarker overall not just associated with one thing going in the right direction, but the net effect? Is it a +5? Is it a -8? If it's a -8, its net score, well, I aim for a lower intake of that nutrient to try to follow the correlation. And that's the approach I've used for the past 10 years. And just using simple blood-based biological age clocks, I mean, I'm doing a pretty good job. So now I do have some weak spots. Everybody's got weak spots. That's why when I hear like Brian or other people say they've got perfect biomarkers, this is impossible. Everybody's got weak spots. Maybe they're just not measured. So, but yeah, I look at as much data as possible. It does get time consuming because if I'm doing 400 metabolites and I've got, 100 dietary variables, just think for each one of those variables,
Yeah.
I'm looking for, so AI is coming that will help with all
Yeah.
of this. But for now, I've gotten pretty far with
Yeah.
just simple unadjusted correlations.
Cool. Well, again, you've got a YouTube channel. It's called Conquer Aging or Die Trying. I think we can probably get the idea of what that's all about. We can check that out. And once again, if people want to consult, how do you do that, Michael?
Yeah, I'm on Patreon. Just come join on Patreon and we can get started from there.
Great. Dr. Michael Lustgarten, thank you so much for being on the show, man. I appreciate it.
Thanks, Buck. Have a good day.
Thanks for listening. A quick reminder that while I am, in fact, a surgeon, nothing I say should be construed as medical advice. Now, make sure to include your physician in any medical decisions you make. And also, if you're enjoying the show, please make sure to show your support with a like, share, or subscribe.